السلام عليكم ورحمة الله وبركاته. Neisseria, Mycobacteria and non-spore forming Chlorine Bacteria. Good morning everyone. Today I'm going to talk about a group of bacteria that previously we discussed what are they and what they claim to do in theory lecture. This bacteria including Neisseria, Mycobacteria, in addition to gram-positive non-spore forming bacilli, which is Chlorine Bacteria. So in this practical sessions we're going to know how we can differentiate between the two genera Neisseria and Moraxilla with the identification of important differences between the two species. In addition to that, differentiate between the two non-spore forming gram-positive bacilli, which is Chlorine Bacterium species and Listeria, to identify tuberculosis, which is acid-fast bacilli, and how we can diagnose this bacteria. Firstly, I'm going to talk about Neisseria. As we know, Neisseria are gram-negative diplococci, contain many commensals. So this species contain many commensals, some of them harmless, inhabit upper respiratory tract and alimentary tract. But this genus contains two pathogenic species exclusively human parasite, which is Neisseria gonorrhea. It is also known as gonococcus, and Neisseria meningitis, which is also known as meningiococcus. Neisseria gonorrhea, which causes gonorrhea in male and female, while Neisseria meningitis causes meningitis, inflammation of meninges. What the general characteristic of this bacteria? Gram-negative diplococci, kidney shape, oxidase positive. All Neisseria species oxidase positive. One of the most important tests used to differentiate between Neisseria species from other bacteria is oxidase test. The principles of this test depend on ability of bacteria to produce endophenol blue from oxidation of oxidase reagent. The oxidase reagent called tetramethyl paraphenylene diamine dihydrochloride. In case of positive result, development of dark purplish color. Within 1 minute, many bacteria other than Neisseria is also give positive result. This slide shows the principles of oxidase test in which bacteria produce endophenol, which is purplish color within 1 to 2 minutes by the presence of oxidase reagent. This slide shows positive and negative oxidase reagent. You can see the positive appear as a blue or purplish color, while negative colorless, no changes in oxidase reagent. This is a mnemonic. Not only Neisseria species can give positive for oxidase. Many bacteria other than Neisseria species can produce or give positive oxidase test. To remember this bacteria, you can use this mnemonic. He loves paint very nice portrait of Papillon AD. H. pylori. L. Legionella. P. Pseudomonas. V. Vibrio. N. Neisseria. P. Pastorella and Procella. Because Neisseria species are closely related to each other, many laboratory tests used to differentiate between them. One of the most important tests used to differentiate between Neisseria species, between Neisseria meningitis, Neisseria gonorrhea, and Moraxilla species is ability of this bacteria to ferment carbohydrate. So the test called carbohydrate fermentation or sugar fermentation, including glucose, maltose, lactose, saccharose. Glucose, maltose, lactose, saccharose. The fermentation lead to acid productions, and by the presence of phenol red, which converted phenol red as indicator in acid detection test. The organism which produce acid from carbohydrate causes a color changes from red to yellow. هالفحص هذا اللي يعتمد على قابلية البكتيريا على تخمير الكربوهيدرات، خاصة الجلوكوز، المالتوز، اللاكتوز. يستعمل انديكيتر في هذه الوسط الزرعي. هذا الانديكيتر هو الفينول ريد. لونه الطبيعي هو أحمر، لكن إذا البكتيريا خمرت الكربوهيدرات يتحول من اللون الأحمر إلى اللون الأصفر مثل ما تشوفون حضراتكم في هذا السلايد. Neisseria gonorrhea, Neisseria gonorrhea, Neisseria gonorrhea ferment only glucose, Neisseria gonorrhea ferment only glucose. While Neisseria meningitidis ferment glucose and maltose. You can take glucose gonorrhea, GG, while meningitidis glucose G and M from maltose. So this is a mnemonic. You can find the first letter of the word glucose from Neisseria gonorrhea, but you can find the letter of the word maltose and glucose from meningitis only. Other tests used to diagnose disease caused by Neisseria species is gram stain. One of the most important tools for diagnosis of gonorrhea caused by Neisseria gonorrhea is gram stain. In male gonorrhea represented by urethral discharge. So, direct swab from urethral discharge are taken, then prepare gram stain for diagnosis the causative agent. In case of gonorrhea positive, gram stain show a gram negative diplococci with a huge number of pus, mainly polymorph nuclear cell, neutrophil. A specimen used in laboratory diagnosis, it is important to differentiate between also causative agent, because in case of gonorrhea the specimen is genital secretion in male, while in female endocervix swab. In addition to that, rectum, thynex, and joint fluid may be also used as a sample in diagnosis of gonorrhea disease. While in Neisseria meningitidis, in case of meningitis, blood, CSF, nasopharynx, and sputum were used as a sample for diagnosis of meningitis. Neisseria gonorrhea in male, as we mentioned, represented by urethral discharge, and the gram stain in diagnosis of gonorrhea in male, it is definite diagnosis. So when you are suspected gonorrhea in male and you take urethral discharge and find a gram negative diplococci in this urethral discharge by using a gram stain, that definite diagnosis in male as a gonorrhea. While in female, gram stain, it is a presumptive diagnosis, not definite diagnosis. Why? Not definite diagnosis in female gram stain, because many normal flora found in vaginal mucosa that resembling in morphology Neisseria species, such as Gardnerella species, and other bacteria. So a gram stain in female, not definite diagnosis, only presumptive diagnosis. And if you suspected that, you must transfer to other step to confirm this result, which is culture technique. هذا يعني انه بحالة الميل الجرام ستين تعتبر definite diagnosis إذا وجدت جرام نيجاتيف ديبلوكوكاي باليوريثرال discharge surrounded by polymorph nuclear cell, surrounded by pus, whatever extracellular or intracellular bacteria. While in female, a gram stain not definite diagnosis, it is presumptive diagnosis. ما تقدر تقول بحالة الفيميل اعتمادا فقط على الجرام ستين انه هذه المريضة لديها جونوريا، لديها سيلان، لانه كثير من البكتيريا بالفاجينال ميوكوزا resembling in shape in morphology to Neisseria species. فبالتالي ما تقدر تميز بيناتهم باعتمادا فقط على الجرام ستين. جوز Gardnerella or other bacteria that resembling in morphology Neisseria species. لهذا لا يعتمد على الجرام ستين كتشخيص نهائي للجونوريا in female cases. This slide show gram negative diplococci of Neisseria surrounding by polymorph nuclear cell. In case of meningitis caused by Neisseria meningitis, CSF sample used or blood used as a laboratory specimen for diagnosis this bacteria. Also, this slide show direct gram stain of Neisseria gonorrhea, urethral discharge, direct urethral discharge by gram stain show gram negative diplococci kidney shape intracellular polymorph nuclear cell في داخل البص cell. Culture characteristics. So the first step in diagnosis of Neisseria disease, whatever the causative agent, you must select the proper specimen to be used according to the clinical manifestation. Then used direct gram stain, and to confirm result, the culture method must be used. And aerobic, this bacteria in culture aerobic require 5 to 10% CO2 for primary isolation. You can use chocolate agar or Thyre-Martin medium, chocolate agar, blood agar, and selective or special media called Thyre-Martin media. So the special media or selective media used to isolate Neisseria gonorrhea or Neisseria species is Thyre-Martin medium. Thyre-Martin medium. The colony are small, grayish, transparent, no hemolysis on blood agar. This figure shows the colony of Neisseria species on Thyre-Martin, and you can see the difference between special media, Thyre-Martin media used for cultivation and isolation of Neisseria species in compare with other media, which is enrichment media, chocolate agar medium. You can see overgrowth of bacteria on chocolate agar, while only Neisseria species growing on Thyre-Martin media, because it is selective media, found many inhibitors such as antibiotics that inhibit growth of other bacteria and facilitating or enhance growth of Neisseria species. This is also a figure show Neisseria gonorrhea on Thyre-Martin agar. Neisseria gonorrhea on Thyre-Martin agar, which is a selective and special media used for isolation of Neisseria species. This media contain many inhibitor substance and antibiotic that inhibit growth of other bacteria and enhance growth only of Neisseria species. In case of Neisseria meningitidis, this table showed the comparative between CSF parameter between bacterial infection with other causative agent. In case of meningitis caused by Neisseria meningitidis, in case of meningitis caused by Neisseria meningitidis or other bacteria, you can see elevated of protein, increased protein concentration, and decreased glucose concentration with increased number of white BC, predominantly polymorph nuclear cell. إذن بحالة البكتيريا meningitis البروتين يزداد، الجلوكوز يقل، وايت بي سي يكون عالي، واكثر شيء الوايت بي سي هي polymorph nuclear cell. while بالفيرال انفكشن نشوف الليمفوسايت يصعد اكثر شيء، الليمفوسايت يصعد قياسا بال ك white BC differentiation. The second bacteria in this lab is Chlorine Bacterium species. There are two species belong this genus. Toxigenic and non-toxigenic. Toxigenic Chlorine Bacterium and non-toxigenic Chlorine Bacterium. Toxigenic, which is Chlorine Bacterium diphtheria. causes diphtheria al-khannaq, causes diphtheria al-khannaq. And non-toxigenic, which is collectively known as Chlorine Bacterium Tuberculosis, Chlorine Bacterium Tuberculosis, Chlorine Bacterium Tuberculosis. Chlorine bacterium gram-positive, arranged in Chinese letter, gram-positive, non-spore forming, arranged in Chinese letter or clumped shape. It's produce powerful exotoxin. This toxin have ability to inhibit protein synthesis by a process called ADP ribosylation of elongation factor 2, which lead to this process lead to destruction of epithelial cell and the mucous membrane that lining tracheobronchial tree. This destruction lead to formation of Gresh pseudomembrane. In more detail we are discussed in theory lectures. But this bacteria, gram-positive bacilli, clumped shape, arranged in Chinese letter. It is contained in highly polymerized polyphosphate granules. It is contained highly polymerized polyphosphate granules called metachromatic granules or volatile granules. These granules can be detected by special stain called Albert stain. So special stain used in diagnosis of Chlorine bacterium called Albert stain. So the first step in diagnosis of Chlorine bacterium diphtheria is by isolating this bacteria. Then to know if this bacteria can produce toxin or not. So to test if this bacteria toxin production or not, we are confirm this result by special test called ELIC test, which is immune precipitation assay, or by PCR to confirm this result. The culture of bacteria by using special media such as potassium tellurate blood agar, Tinsdale agar, or Loeffler medium. So this is a special media used to diagnosis of Chlorine bacterium diphtheria, either potassium tellurate blood agar, Tinsdale agar, or Loeffler medium. Of course, in addition to that, we are used special stain called this Albert stain or Klebs-Loeffler stain. Albert stain or Klebs-Loeffler stain. A special stain used in diagnosis of Chlorine Bacterium species in addition to culture by using special medium such as potassium tellurite blood agar, Tinsdale agar, low flowers medium. And this is elictase and P complex and diverse cell wall cell wall composition. This cell wall composed from peptidoglycan intermingled in a matrix of protein, carbohydrate, and lipid. And as we know, lipid fractions contain three types. Lipid fraction in cell wall of Mycobacteria contain three types: cold factor, wax D, and mycolic acid make the cell wall of this bacteria impermeable to gram stain, so it is not easily to be stained by using a gram stain. In this case, we are used a special stain called acid fast stain. So the diagnosis of TB disease either by skin test or chest x-ray, while in case of laboratory diagnosis is in case of pulmonary tuberculosis, we are take a sputum and used either Zell-Nelson stain, acid fast stain اللي راح اليوم تشتغلوها بالمختبر، بالإضافة إلى أكو other special stain هي fluorescent stain تستعمل عادة في حالات البحوث يسمونها أورامين رودامين تيست. هاي fluorescent stain يعني حالات البحوث فقط تستعمل، لكنه most common and popular stain used in diagnosis of tuberculosis was Zell-Nelson stain أو AFB stain, acid fast bacilli. In addition to that, greenish color of Lowenstein-Jansen medium as a result to the presence of malachite green, which is one of the selective agents found in Lowenstein-Jansen medium to prevent the growth of other contaminant or other bacteria. إذن هذا اللون هون اللي تشوفوه حضراتكم باللونشتين-جينسن ميديا سبب وجود صبغة خاصة هي الملكايت جرين. So this is a TB culture on special media, Lowenstein-Jansen media, rough, tough, and buff bacteria, colony, non-pigmented. Other test used now, it is widely used in Iraq, called gene expert MTB rifampin test. This test simultaneous rapid tuberculosis diagnosis. This test called gene expert, it is diagnosis and at the same time rapid antibiotic sensitivity test. So it is gene expert test, it is diagnosis for tuberculosis, and at the same time it is rapid antibiotic sensitivity test. Rapid antibiotic sensitivity test. It is used to contributing contributing the rapid diagnosis of TB disease and drug resistance at the same time. جين اكسبيرت الآن موجود في العراق ويستعمل، وهو واحد من الفحوصات اللي يوصى بها في تشخيص التي بي وكذلك في دراسة الريزيستنس. هذا هو الجين اكسبيرت، هو simultaneous rapid tuberculosis diagnosis test. It is diagnosis and at the same time used to detect sensitivity test. يدرس إذا كانت هاي البكتيريا sensitive or resistance للأنتيبايوتيك، خاصة الفيرست لاين أنتيبايوتيك. اليوم انتم راح تشتغلون الزلنسنستين اللي راح تشوفونها بالمرفق مال الفيديو. هاي هي special stain for diagnosis of Mycobacterium tuberculosis. تتكون من 3 reagent: strong carbolfoxine, acid alcohol and malachite green or methylene blue as a counter stain. اليوم راح تستعملون هو الميثيلين بلو. إذا زلنسنستين تتكون من 3 reagent: carbolfoxine, acid alcohol and methylene blue. acid alcohol and methylene blue. The principle of this stain depend on if the bacteria are Mycobacterium tuberculosis, so it is resistance and return the primary stain. Resistance to decolorizing, which is acid alcohol, and it's have ability to return primary stain carbolfoxine. So it is after decolorizing, it is return carbolfoxine and still red color, while in case of it is non-tuberculosis bacteria, the acid alcohol decolorize the primary stain, which is carbolfoxine, and converted the bacteria colorless. Then when you added methylene blue, the bacteria take this counter stain. So this this figure show Mycobacterium tuberculosis by Zell-Nielsen stain. It is red, slightly curved, straight shape, and sometimes straight shape. Also, this is a Zell-Nielsen stain, Mycobacterium tuberculosis by Zell-Nielsen stain. You can see a red color, slightly curved or sometimes straight bacilli. This is acid fast bacilli or Zell-Nielsen stain for detection of Mycobacterium tuberculosis and